can see the screen.
Yes.
So I'm Gordon Shepard, and I want to start by introducing the team. So I'm at the Department of Neuroscience at Northwestern on the Chicago campus, and I think John Barrett and Man Gao, who are postdocs and in my group, are with us on the call as well. And they listed here are some others in my group, as well as in my department who are involved. On the upper campus in Evanston, I want to highlight Mitra Hartman, who's a roboticist in the engineering department, and her group, who are helping us with some of the instrumentation and technical aspects. And then we have other collaborators, Arlette Colta at the University of Montreal, who's an expert in mastication, and Minghong Ma and Dan Wesson, who are experts in olfaction and sniffing, who are also helping us on these disparate aspects of this behavior that we study, which is oromanual food handling. And the goal of this project is to do multimodal analysis, recordings and analysis of this behavior in freely moving animals. And so I want to introduce this by explaining what we mean by oromanual food handling and why we think it's interesting. So in general, animals handle food as this very critical stage between going out and foraging for it and procuring it, and ingesting, consuming, and ultimately digesting the food to gain nutrition and energy. And so many animals, of course, don't use their hands to do this. Most ungulates, dogs and cats to varying extent do have a little bit of hand use, but primates, probably all primates, use their hands extensively to handle their food. And really many rodents and a number of marsupials do as well. And so we view this as a very ethological and survival critical form of goal-directed free object manipulation. And we think it's interesting from a behavioral and motor standpoint because it's a very complex form of dexterity involving not just the hands, but also the head. And as I'll show you, some very other disparate motor systems. And just to mention, Paul Chisek has recently written about this, about the general concept of handling probably being a major driver, certainly for primates, of brain behavior relationships and evolution, something we certainly agree with. Down here in the bottom, this is just an extremely schematic breakdown of some of the basic movements, kinematic movements that we've identified from studies that John Barrett has pioneered in my group with head-fixed mice. And so there are these different components. There's gripping and regripping and biting, holding and chewing. The one in the interest of time that I'm going to focus on today is sniffing of the food. If I had more time, I would love to tell you about the fascinating cranial morphology of rodents and how that puts constraints, specifically that rodents can only either bite or chew, but not the same at both times. And that shapes many aspects of the whole behavior, actually. John has just published a paper on this. So what I want to do is introduce you in this short video here of a squirrel to what it looks like when rodents oromanually handle food. So I think I'll just play it again. I think you can appreciate that this wild squirrel is holding the food between its thumbs. And not only that, those thumbs actually have flat nails instead of claws, which they have on all the other digits. And this turns out to be true of mice as well. And so now I'll show you some movies. This is of
1 minute.
A mouse that is head fixed and consuming a pellet of food. This is at normal speed, and it's a bit of a blur of activity. This was obtained with a camera running at 1,000 frames per second. And so if we slow this down, we can see some of these basic elements of movements underlying this overall complex behavior. And so here is the transport to mouth movement that is very characteristic and starts off a bout of this oromanual manipulation at the very active movements of the hands and the mouth before the food is lowered down again. And I think in the interest of time, I'll just skip ahead and show some regrips. These are very rapid adjustments of one or both hands to get a new grip on the food as it's eating it up at the mouth. These are extremely fast. They're over in less than 20 milliseconds. I think that's why they had been missed previously and required very high-speed recordings to detect. This is a breakdown of some of the goals in the grant. The overall goal, as I said, is to basically record and analyze everything we possibly can to understand this behavior in depth in freely moving mice. And since time is short, I'll just dwell on one example of where we've made particularly much progress on this. And so this is Mang Gao has set up this camera system here. This is a robotically controlled camera on the bottom. It's under the control of a top camera that has a wide view and is feeding the signal to Deep Lab Cut Live, which is trained to detect the head position and direction and sends a signal to this XY translator on the bottom so that we can get a zoomed in close-up view with the bottom high-speed camera. And this is what it looks like down in panel E. And here we're focusing on understanding this sniff behavior that I mentioned. And so I'll show you examples in a second, but basically these are also extremely fast movements where the mouse quickly puts the food right under the nose, just extremely briefly. And so to understand this really well, we need to understand not only the kinematics, and we do that by extracting the hand to nose distance from the Deep Lab Cut recorded or tracked kinematics. That's shown in blue here. And so these downward dips are these food sniff events. In the green is this beautiful respiratory signal that Mang has managed to record. So this is the breathing signal going on. And what we want to understand is how they're related during these food sniff events to address, are these actually sniffs, or is it just, there are various possibilities shown in panel B here for how these may or may not be coordinated. So here's an example of these food sniff events. We actually have much better videos at sharper focus, but this exemplifies it very nicely. And there are two sniffs that happened in succession. Here comes another. You can see the peak sniff event marked by the red dot here. And this is at normal speed. And if we now slow things down, I think you can begin to see how we can make more sense of what exactly is going on here. So here's one sniff movement, again, marked on the bottom by the peak of the kinematic movement. The bottom is the thermistor or breathing signal. The top is the hand to nose distance. So what you can see is that during these two sniff events, there was a very brief, I didn't mention, but inspiration is downward in the breathing signal. And so in both cases, and in the previous one as well, there was a very brief inspiratory sniff, right timed almost with millisecond precision to when the food is maximally close to the nares, right under the, placed right under the nose. When I rehearsed this, I kept running out of time, so I put this here in case we run out of time now. This gives you not a bird's eye, but a board's eye view of what the actual apparatus looks like. It's a bit of a bird's nest down here, but this is that box. Again, here here's the team. I do have more videos I can show, but I assume I'm out of time.